So, yesterday in my talk you have seen we have covered like brain development and its implication for atypical development, particularly in a disease like neurodevelopmental disorder like autism. What we are going to talk about is something which is much more interesting according to me, because this actually goes into understanding whatever you learned in this workshop at the previous stages. How do you do actually modeling of the brain in order to understand the mechanisms which underlies actually all types of computations. So, first questions that we asked in our lab is to understand what is the structural or functional alterations that actually accompanies the aging process. When I say aging by I means like when we are looking at participants in the age group of, let us say, something like 18 to 20 years to all the way to something like 80 years, and either longitudinally or cross sectionally to find out essentially whether there is change in the underlying neuroanatomical connectivity, which you have heard about from many of the previous speakers' talk in terms of the diffusion tensor imaging data, as well as from the functional MRI fingerprints, and as well as from the brain dynamics. These you can do in animal models as well. If you can study animals with different ages, like for example period of gestations, few weeks from their birth all the way to when the animal die, right. Ideally the brain should undergo a lot of changes over that period. These changes could be at various scales. As you can see, there could be change in the gray matter volume, there could be change in neuronal size and synaptic density, there could be change in white matter, and in our lab we are particularly focused in these white matter changes and white matter insults in the brain as you become older, right. And what does that has an implication and effect on the emerging brain dynamics or the brain network dynamics. There are other things which can also happen structurally, such as cortical thinning or regional atrophy or dopamine depletion. If you are interested in molecular neuroscience, some of these may be of huge interest in order to understand essentially what is the impact on the brain dynamics. And there are also cognitive changes. The cognitive changes that particularly we focus on in the lab is related to attention and working memory, but that is not the, yes.
So what do we mean by dopamine depletion?
So the dopamine actually concentration changes in the brain.
Production of dopamine.
Yeah, but I don't want to get into that. That's not the primary goal of my paper or work, right? Yeah. Yeah, I don't want to give you an insight because I don't know much about it. Yeah, myself. So the recording techniques that we use typically in neuroimaging lab across the globe, by all labs I mean, is using EEG, MEG or MRI or DTI. These are standard techniques. You have heard about it several times during these workshops. These are the go-to techniques when you are talking to a cognitive neuroscientist who is actually recording brain activity, particularly from human participants, right. And then you can look at some of the analysis techniques, and Arpan obviously helped me a little bit in the previous talk by discussing about spectral methods when he was talking about auditory cortex and looking at the data that was collected during behavior, right, using EEG. And there are spectral analyses, there are functional connectivity measures, there are structural connectivity measures, and there are behavioral studies. But we are also interested in task fMRI, right, when the subjects are doing some kind of task like an attention task or a goal directed attention task like Sridhar mentioned about, or for example like Arpan mentioned about, specifically related to auditory attentions and others. So if you look at the STARS data, you may ask the questions that what is the most interesting observations that you get from the aging data. And the most interesting observations that you see is there is a very interesting shift from the posterior areas, which you have heard about, right. Most of you remembered by now where is our occipital areas or visual areas, which allows us to see the world, right. And it seems as you become older there is a flip. So the information processing is taken up by anterior areas, and some of the earlier evidences which observe these in fMRI signals, they call these posterior anterior shift, with aging, which seems to be a very important cognitive neuroscience theory of the brain aging, or an observation that actually associated with aging process. And when we are talking about aging, we are not necessarily talking about like MCI, dementia, Alzheimer's disease and those kind of things. This is like normal healthy aging process where you observe these differences. Second observations that people have often reported, which is called hemispheric asymmetry reduction in the older adults. You have heard about actually structural asymmetry in the first talk, and also you have heard about asymmetry in speech and music processing, right. So, here if the individuals are given some task, something like a word pair in a queued fashion or in a queued manner to recall the word pair, which is more like a verbal memory task, right. So, when you are asked to do a verbal memory task by encoding the word pairs and then later recalling it, right, after some time, the younger adults could carry out the task actually by using only one single hemisphere, whereas the older adults require actually recruitment of brain areas which involves both the hemispheres, both the areas. So, that goes to show that there is more distributed processing as you becoming older, and one could possibly interpret as like a compensatory mechanisms that kicks in because the earlier structures are no longer reliable as you are becoming older, or one could interpret these something like as a more integration among the brain areas rather than more specialized and segregation or segregated manner of processing. Okay. If we go further, we also see there are theories which talked about these compensation phenomena, which is called crunch theory, CRUNCH, which is called compensation related utilization of the neural circuits. That means, if there is like a specific kind of task in which younger individuals are able to only use their left hemisphere or right hemispheres in order to carry out that task reliably, older individuals always needed essentially bihemispheric activations in order to carry out the same task. And finally, by putting all these three ideas like PASA, Harold and Crunch, these models together, cognitive neuroscientists actually talked about what is known as scaffolding theory of aging and cognitions, though they said that when you are doing a verbal working memory task, that means words you are remembering and then you are trying to recall those words from your memory, okay. In those cases, you have more frontal bilateral activity in older adults during the verbal working memory task, and in older adults you generally see higher performance in long term memory task rather than short term memory task. So, these are very interesting set of psychology based studies using functional MRI and cognitive neurosciences evidence, which points towards different types of neurocognitive theories. However, if you fast forward these to modern times, right, because these studies were carried out back in 2009, 2010, and now we are in 2025, and if you fast forward, you see there is a different way of presenting the same data. What people are talking about, what matters in the brain is the geometry in terms of information processing. So, brain is basically essentially a neural field. It has a spatio-temporal structure. It has neurons which are packed in a volume, right, in order to carry out a task. If the brain size was much bigger, like a size of a football ground, right, it would have been very different than what we have. So, evolution has created actually this kind of a brain in which every structure has been packed in a particular type of geometry, and that geometry actually constrains the human brain functions and the kind of brain dynamics that is generated from that structure, right. And we also argued, or at least we argued, there is a huge amount of contribution of geometry to these human connectome, and these topological organization, just like you saw in my previous talk yesterday, the topological organization, like Arpan mentioned about modularity, hierarchy of information processing, they must be very critical, right. and using those ideas one can essentially try to find out how does the functional connectivity and the dynamics actually changes as you become older, and what way it differs from the younger individuals. Okay? And we proposed back in 2017 an idea to track those dynamical changes based on a measure called metastability. Metastability is a way to think about when the brain is not fully at synchronization, that means the neurons are not completely synchronized with each other, but they are trying to go to synchronization, but some things are restricting them to get fully synchronized. So, these like stability of going into synchronization versus not synchronization is a process which is called transient stability or metastability, which shows you the variations that can be measured from empirical data or observations. So, we are interested in a little bit of metastable brains as people become older. In fact, we argue further healthy aging is nothing but a search for a new normal, because we are no longer normal, right? When we are born, we have a different geometry. When we are older, we have a different geometry. So, therefore, what Robert Browning said is the right thing, that grow old along with me, the best is yet to be, the last of life for which the first was made. And we know these, if we translate these into neuroscience, the areas of the brain that develop fast, they go into neurodegeneration fast, right? Okay? The ones which are made first, they go into neurodegeneration last. That is the reason why many people get into memory related impairments as they become older, because those are the brain areas supposedly develops very, very fast in the developmental process. So, therefore, the brain aging one way can be thought about by using these whole brain computational models that we call WBM, which is the latest development in the literature. So, some of you can actually read about these whole brain computational models. They can be combined with neuroimaging data, and that gives you a very powerful recipe in order to tackle these questions. To understand them in the concepts of metastability, which is a theoretical framework that can systematically account for the variability or observed variability in the brain dynamics. How you do that? I will show you. So, if you look at the data that we compiled, what we see over here, can anyone tell who is in the audience just by staring at it? Some of you are coming from a quantitative background, also from physics or mathematics background. So, you should be able to, irrespective of your knowledge of biology, does not matter whether you studied biology at some point or not, okay, you still can interpret the data, right. So, what do you see which is the most critical observations over here? Yes?
Sir, I have observation for the last panel.
Yeah.
Like when the brain is young, it is more constrained to one place, and as it ages, it gets more and more randomized.
Okay, that's one observation. What do you see in the first row actually, that first panel? Do you see any specific pattern?
See the X and Y.
So, the y-axis is called number of fibers and the x-axis is the age in years. So, when we look at the number of fibers as a function of age, we see there is a systematic decline, right, with age. The number of fibers are progressively actually going to decline and reducing as we become older. But when we look at the functions, right, we see that there is a increase in the functional connectivity, the mean functional connectivity as a function of age between particularly the well reported resting state networks in the brain, whereas there is an opposite trend when we look at basically within resting state networks, and that also happens because the number of modules that you see for the younger individuals seems to be actually like much less compared to elderly individuals, which means that these modules are getting broken into creating new modules. These new modules are created for the elderly individuals, which are actually kind of, is a process called in biology de-differentiation. So, they are getting differentiated more and more, which kind of supports that idea that the brain is no longer having those specialized set of units or networks to carry out those meaningful tasks, but it requires essentially the recruitment of several of these modules in order to carry out possibly the same task by the younger peers, right. The second thing you observed in structural connectivity is that when you go from 9 to 20 years to 70 to 85 years, you see much of the between hemispheric connectivity, that is long range connectivity or interhemispheric connectivity, is actually going down, which is something which you are talking about as the randomization of the brain network. So, these loss of interhemispheric versus intrahemispheric connectivity is something which is very important, and one can study the ratio of the intrahemispheric and the interhemispheric connectivity and to what degree this has actually changed over the course of lifespan, yeah, and use that as a parameter in order to understand what is the implication of that in terms of the functional connectivity and also brain dynamics. Now, we take a physicist approach because my background is physics, okay. So, we take a physicist approach to ask if all these things are happening, can we find the neurocomputational principles which gives you a balanced excitation and inhibition, okay, or balance between excitatory neurons and inhibitory neurons, and the balance between these EI can give you some kind of understanding of the brain dynamics. Secondly, is there a way like we can bridge the two scales, one scale where we are having these observations, they are macroscopic scales, and the other scales from which the cellular dynamics changes is the microscopic scale. So, can we have a model that integrates these macroscopic scale or mesoscopic scale with the microscopic scale, and in that way we will have an understanding of, I do not know, Professor Upi Bhalla, if he talked about like moose and those kind of models, right, these multi-scale kind of models where you can actually have like both the molecular dynamics or the cellular dynamics as well as the brain dynamics that you observed at the level of population firing rates, right. So, Arpan myself has collaborated a lot in these for long period of time with some of Arpan students and also my students and postdocs, including Priyanka who is here. We have developed actually a very interesting set of framework, and I am going to discuss in remaining part of the talk actually about that framework to address this question of biological aging and the complexity that you observed in the dynamics, and how do we find out a way to see what get essentially compensated as your structural connectivity deteriorates, as we have seen our previous slide. So, if you want to aptly summarize what neuroscience actually is doing all these years, this figure is probably one of the best figures which summarizes these at various scales or across scales. So, you can have a phenomena which is at the cellular or subcellular level, and as a biologist you may be very interested in that. If you are a systems neuroscientist, you may be very interested in population level or at the level of the neural ensembles, and if you are a cognitive neuroscientist like me, you may be very interested in the level of the brain regions and brain areas. These are spatial scales separated by many centimeters and also level of organizations. If you go along the lifespan, you can also look at essentially the developmental time scale, and you can observe these developmental time scale which tells you what happens from now to next 5 years versus what happens from now to next 50 years, right. So, this is also called temporal scale, what is happening actually to the brain dynamics and the brain network reorganization as we look at basically the temporal scales. And the third possible scale is the topological scale. The topological scale is what we were talking about yesterday about modularity and like hub and degree centrality of the brain networks and the nodes. So, you can have a very local organizations to all the way to you can have a very global wiring between the neurons, And some of these wirings in the network could follow even small world network topology, right, okay, or other types of network topology, which is in some way is the geometry that constrains the brain functions. All three of them, capturing it is a humongous task and very difficult to set up actually experiments to observe these phenomena which is changing across spatial scales, across temporal scales, and also across topological scales. However, we are more interested in macro scales, which is dealing with these cognitive systems, and we believe actually, as many other groups or labs across the world, is the interactions among dense web of connected neurons that give rise to vast repertoire of cognitive functions, be it language, be it consciousness, or any other hierarchical complex functions, which may not be even present in other animals, but humans have it, right. So, these actually comes from the interactions among dense web of connected neurons giving rise to vast repertoire of cognitive functions, but these may be a very highly neurocentric view, and there, while we talk about or neurons and their interactions, there may be other cell types as well, like astrocytes, glia, or oligodendrites, which may play an important role in computations, but by far the evidence that exists in neurobiology, it predominantly hints at neuronal interactions at various of these scales that I am talking about. When we ask the questions that okay, fine, we can have a model which looks at basically the interactions among the densely connected web of connections among the neurons, but what is you are looking at, what is that you are looking at. So, if you look at the correlations between pairwise correlations between brain regions, this is something what you will be interested in. Okay. So, when you are asking some participants go inside the scanner and you are asking them to remain vigilant and not think about anything particularly, because you are not giving them any goal directed task, except you are asking them not to sleep inside the scanners, and if you record their brain activity, this is exactly what you will observe. And this is not for one single individual or a participants, you can do these on thousands and thousands of individuals. You will always see this kind of patterns which is observed in the brain, which are known as spontaneous correlations. These spontaneous correlations are recorded in rodents and also in C. elegans nowadays. Okay. It is not just for humans, because the spontaneous correlations gives us two ideas. One, it gives us an idea that whether the brain is going through the same processes as it does during wake time while it is processing the environmental stimuli. Secondly, whether we have a very noisy baseline, right, a baseline activity which is sort of like driven by neuromodulators, right. For example, when you go into sleep, the acetylcholine concentration go up, that actually puts you into sleep. So, the acetylcholine becomes actually a key neuromodulator or a driver. Similarly, when you are in the awake state, there are several neurotransmitter and neuromodulators which may be actually driving these activities. So, we are not interested or not able to infer from here the causal relationship, but we can always get the correlation between, let us say, this green time series signal and the red time series signal, right, which are recorded from two different brain regions. Now, if this is our data, this is what we want to actually model using our approach, we can go for it. Secondly, we can also look at the amplitude in terms of the Z scores for different of these brain regions and find out the maps of these brain regions using independent component analysis, where we can look at actually the visual areas or different sub areas of the visual areas, including fusiform gyrus, which is important for face recognitions, which is lateral occipital cortex for object recognitions. We can look at default mode networks, which is actually implicated in self related processings. We can get auditory networks, which Arpan was talking about in his talk. But how come these networks are activated during spontaneous activity when you are not doing any specific task? And the answer is that some of the time when we are thinking about some face, automatically those areas are strongly represented. When we are hearing about some, or when we are thinking about some sounds, the areas of the auditory cortex is also kind of activated, right? Yeah. And this happens often with us, specifically when we are doing mind wandering, right? Yeah. When we are off the task. And these kind of spontaneous correlations therefore is important. So, we looked at 3 different data sets, one which came from Cambridge University aging neuroscience study, second which came from New York NKI Rockland data, and also the third data that I myself were involved in collecting the data in Berlin, Germany from Max Planck Institute. Okay. So, using these 3 different data sets, we wanted to find out and do test retest validations to see whether the models can predict in all 3 data sets the same kind of empirical observations, right, or experimental observations. And we had similar number of distribution in terms of gender, age and various other things that was controlled for. None of the participants has any cognitive or neurological deficits in order to first look at their data. And using these data, we looked at first their anatomical connectivity, which is estimated from diffusion tensor imaging. So, if you look at these matrix which are called SCs, SC is not superior colliculus by the way, SC is structural connectivity. Structural connectivity is estimated on a matrix where the part of the connectivity which is actually showing you very dense connectivity over here, this is one hemisphere, this is another hemisphere and this is the interhemispheric connectivity. Okay. So, if you think about brain is a 3D structure and you want to flatten it on a piece of paper, this is how the structure will look like, right. So, these areas are strongly connected, but majority of the areas in the brain are very sparsely connected, right. That is how our anatomical connectivity is. And when you compare the young versus older subjects, you do find that there is a altered reorganized functional connectivity. You have heard of course like about symmetry of connectivity from Sridhar also in yesterday's talk. So, what we are interested in, to what extent SC can predict FC, right. Underlying structural organization can predict functional connectivity, and do you see any kind of changes in the underlying structural connectivity when you compare the 2 groups, the younger group with the older group, right. Yeah. And what we find out that there is some preservation over here. The preservation is in the functional modularity, although there was change that was observed over here in terms of altered reorganization in FC by looking at the brain areas and their connected nodes, right, in terms of the age and their strengths, the number and the strengths. There was no change. You see, this is non-significant. There is no change when you go from the young to elderly individuals in terms of their modularity, particularly in the functional reorganization, although there is a change in the structural connectivity. So, one question that comes is what constitutes actually these functional reorganizations to the extent that this modularity is preserved, or the functional modularity is preserved even when you are becoming older, with variety of loss of connections that you observed at the structural level. So, in order to address these, Shuman, who was a postdoc with Arpan and myself and also Priyanka, they both together came up with these modeling framework where they asked about, can we have a computational pipeline to relate the GABA and the glutamate, which are the 2 major neurometabolites, and to look at their concentrations to find out whether we can predict the brain functional connectivity that you saw in the spontaneous activity, spontaneous correlation, and also the brain dynamics. And the way it goes is that you start from the molecular scale, which is in nanometer, right. Okay. where you have neurotransmitter regulations to all the way to the synaptic scale or at the single synapse resolution, to mesoscale, which is the brain regions, finally to the whole brain models, that is actually a large scale model. What is your intent to observe is to find out how does the metastable brain dynamics, that is basically deviation from synchrony, actually change in the brain, and can you actually predict that, and based on what kind of GABA glutamate ratio you should be able to predict that. Okay? And this is what we investigated. Then we looked at the modeling part, where we can actually derive a model based on the previous works that exist in terms of understanding the whole brain computational models. The second interesting thing that we had in mind is to look at the outcome from these mean field dynamic mean field model or multi-scale dynamic mean field model. So, the model is called MDMF, multi-scale, because it can bridge the two scales as we have seen, and it is a dynamic model because it can actually produce the dynamics of the mean field activity of the neurons. So, using this model, can we vary or change the two free parameters, one is GABA concentration and second is glutamate concentrations, and see the outputs of the model which is interpreted across healthy aging, and compare that with the experimental observations. So, if we can, then the simulated data or simulated functional connectivity from the models can be fitted with the experimental functional connectivity, and we can predict the same kind of principles of segregation and integration which Arpan was talking about actually in the previous talk between brain areas, and quantify that, and we can look at the qualitative differences between the predictions based on the metastability that is generated by this model. So, when we look at the experimental data, we measure three different informations. One is the network segregation, second is the network integration, and the third one is the network resilience. What we find that in structural connectivity you see these increase in the modularity, which is statistically significant, whereas in the functional modularity it is not actually statistically significant. And then there are various measures like transitivity properties which also show very high significance when you look at the functional connectivity data. You also find in terms of the network integrations, functionally it is actually increasing, which kind of confirms to the previous hypothesis which we talk about, that as brain becomes older there is more creation of new modules, and these modules may be interacting with each other, giving rise to an increase in the overall integration properties of the brain. And that is what we see in terms of measures like global efficiency and average characteristic path length, which can be all the different network measures that you can apply. If you do not know about these measures, we can discuss offline and to explain to you, but I do not have a slide to explain each of these methods separately right now, but we can certainly talk about these. But these are the very rudimentary network analysis methods that people used in network neuroscience in order to quantify the level of integration that is happening between brain regions. And similarly, we can look at network resilience by measuring an index which is called assortativity and node betweenness centrality. I believe I have 10 minutes, right, at least. Yeah. So, now my argument is supported by many of the studies that exist from ages in neuroscience. So, there are broad agreement among all neuroscientists. There are three levels of organization. The first level is a biophysical level, which is actually microscopic, as you can see over here. The second level of organization is single population level, which is actually mesoscopic. So, for example, if you give a task to a participant to tell what is this picture, this is nothing but Sydney Opera House, all of us can recognize these. The participants will probably correctly respond to that task, but which part of the brain is actually processing these information? It is actually the visual neurons, which most likely through their population activity at this intermediate level is carrying out essentially an interacting population activity that is giving rise to the perception of this, right. So, you are looking at essentially at this 20 micron level where you have the mesoscopic organization, and what I am talking about right in today's lecture is at these macroscopic observations, that is even higher level where you require a different set of models than population models or biophysical level. But what is interesting about this MDMF that I talked about, that we could in some sense go from the biophysical level to the macroscopic level by incorporating these parameters where we can look at the effect of the GABA and the glutamate ratio and their concentration, right. So, this is what we do. There is a whole series of like body of work from our collaborators, various authors, and also our lab itself over the years. So, you can check out some of these papers which discusses about detailed spiking neural networks like Hodgkin-Huxley and other types of models that can be like implemented in each of those areas of the brain. And if you have a structural connectivity, which is the connections between one cortical area and other cortical area, and if you apply these neurodynamical model and embed them in each of the brain regions, they can produce actually in a bottom up fashion these functional connectivity. Now, you can fit this functional connectivity to the experimentally observed functional connectivity, which is coming from resting state networks, for example, which looks at the correlation of the Bolt signals, and various works, tons of works have shown these to be highly reproducible. There is no ambiguity as to these is concerned. The other thing that we are interested in, what Arpan talked about, is synchrony and metastable brain dynamics. So, where you can think about individual nodes, but you do not talk about these in terms of like a dynamical mean field model or a spiking neuron model, but you think about the brain as kind of bunch of oscillators, where these oscillators have individual phases. When the phases are locked with each other, this is called phase locking phenomena, which is also called phase synchronization in mathematics, right, which requires a certain type of parameter that allows these phases to actually get synchronized with each other, or when they are deviating from the synchronization or showing a lot of standard deviation from the synchronization, that gives rise to what is known as metastability. Our conjecture was that probably when you are looking at aging brain data, you are seeing less synchronization, but you are seeing more a tendency towards maximizing metastability, because by maximizing metastability, it allows the brain to kind of compensate for all the structural loss and changes it has undergone. So, in order to do that, we first simulated the DMF model, not the multiscale DMF model. Okay. One is a DMF model, another one is a multiscale DMF model. So, this is a DMF model which actually allows you to connect each individual brain area with a certain set of excitatory neurons and inhibitory neurons. You can see 200 neurons are there per area, multiplied by 66 area that you have actually derived from the diffusion tensor imaging data, where you know the connections between these 66 areas or the structural connectivity. Now, if you let this model run, okay, this is a mean field model which has long range connectivity, like long range connectivity, and then you have recurrent connectivity which is within the brain area, right, and this is self inhibition and this is self excitations. So, if you run this model, this is the kind of bifurcation diagrams that you get, where you find essentially the model produces a very boring spontaneous state where most of the neurons remain in the low activity state around 0.5 hertz or 0.6 hertz and those kind of stuff. And then if you like change the coupling strength to higher values, then the neuron starts producing actually a very rich landscape, showing high firing rates or low firing rates, which is typically observed in the brain data. Okay. These models have been very good in terms of being very parsimonious model of producing brain dynamics data, and with a lot of explainability in terms of like how does it work, where this G is basically the global coupling strength between many different brain regions, and as you scale up the G, you are scaling up the excitatory inputs to the different brain regions. And you can actually go for a simpler version of this model as well by doing some kind of linear approximation on the transfer function of the inhibitory neurons, which typically fires between 8 to 15 hertz. So, you can have a Fi curve which is almost linear, and based on that assumption you can remove the inhibitory population practically from the picture, only deal with the excitatory neurons, and you can still reproduce much of the brain activity. And there I think you only have limited amount of dynamics that you can observe. The last model that I am going to talk about is basically a balanced network where you have a perfect balance between excitatory and inhibitory neurons. How many of you know how many excitatory neurons and inhibitory neurons we have in the brain? I mean, what is the ratio typically in a given brain regions? 2:1? 3:1. You have never wondered, right, why it is 3:1, right? So, if you look at the previous slide, it says that the inhibitory cells typically fire 8 to 15 hertz. Of course, this is an empirical observations, there is a lot of variability across it. But the thing is, because the inhibitory neurons fires almost 3 times the excitatory neurons under spontaneous conditions. So, therefore, in order to preserve the balance, the ratio between the excitatory and inhibitory neurons are typically found to be 3:1. Okay? And that is basically the balance we are talking about. So, when it is in the balanced state, then in that case you can actually produce a very interesting dynamics, where half of the time with coupling strength varying between 0 to 4, it can stay actually in this low mode, right, low attractor solution or fixed point attractor solution, where you get basically a very boring firing rate which is around about 3 to 4 hertz, and then you get essentially a very high firing rate when you go into an unstable regime. Okay? So, these dynamics actually prompted us to develop this model by one of the postdoc in the lab, Amit Nashkar, who was working with Arpan, and who developed this model by showing a current based multiscale dynamic mean field models. I am not going to go into the very big details of it, but want to highlight the most important points, which is basically these T GLU and T GABA, which are the glutamate and the GABA parameters that were introduced in the model. The first 2 terms are the input currents of the excitatory neurons and the inhibitory neurons. The second 2 terms are the firing rates of the excitatory neurons and inhibitory neurons, typically follows a sigmoid curve or a sigmoid functions to kind of estimate the firing rate of the neurons. All the other parameters which are biophysical parameters are derived from biological experiments. So, they are fitted into those parameters, and the last 2 is basically the synaptic gating function, which actually closes and inactivate the synaptic gates essentially whenever there is a change in the firing rate of the neurons. Okay? And these whole brain dynamical model can produce what we observed previously in the experimental data, that is the network segregation effects, network integration effects, and also the network resilience effects by appropriately tuning the concentration of the GABA and the glutamate parameters, and showing that there is a difference between the glutamate values of the younger and the older individuals, and there is a difference in the glutamate and the GABA ratio that primarily actually drives the changes that we are observing in the whole brain reorganization of network properties as well as the dynamics. The second thing that we were very interested in is to see the metastable brain dynamics, which is sort of like reported by other studies as of like brain waves. So, if you let this movie run where each of the brain areas are connected by nonlinear dynamical models in this fashion, which was shown by Michael Breckspear, and you have the connectome, then if you look at the spatiotemporal brain dynamics for the aging individuals, you kind of see there is a difference in the metastable waves and the metastable patterns, which is generated based on the connectivity which is constrained for the younger individuals and the elderly individuals. Elderly individuals seems to have like very different kind of state switching and different kind of connectivity, functional connectivity and metastable dynamics. If I let it run one more time, you can see the pattern between the aging individuals is very different from these individuals, which can be, or which we have also models using something we called spectral graph models, which actually allows you to see how does the diffusion process changes in the brain as you become older, okay, because it's a diffusion phenomena as well. So, now going forward, we can model that dynamics what you saw in that movie by looking at 18 years old all the way to 80 years old and ask the question, if in the two parameter space, that is GABA and glutamate, the concentration is adjusted or changed in the mean field dynamical model, right, or multiscale dynamical mean field model, then do we see actually a systematic change in the maximal point where the metastable dynamics becomes maximized, right? And we do call this kind of parameter space in physics as phase diagram, okay. So, phase diagram something which you may have heard about, where like the same element can become a water or ice. These are called points of isotherm, right, where there is a phase transition. So, this system also undergoes a phase transitions, and you can kind of see an asymptote which is changing actually for the very young individuals all the way to like elderly individuals, and you can find essentially that is primarily or predominantly happening because there is a systematic decrease in the glutamate sort of concentration between the young and the elderly individuals as well as the glutamate and GABA ratio. So, we can keep on doing these for different values of the T GABA and the T glutamate parameter that you saw in the MDMF model, and find that for all the data sets that is used for this study, whether it is Berlin data or NKI Rockland data or CAMCAN data, some of these results are reproducible. Secondly, we can also estimate the optimal value of the glutamate and the GABA, because this is really an optimization problems, because the question that you are asking is at what GABA and glutamate ratio or at what GABA and glutamate concentrations the brain dynamics is maximally metastable, right. So, therefore, it is an optimization problem, and if you can optimize those values, you can find out subjects wise alteration in GABA glutamate concentration or ratios, which can be inferred from the whole brain dynamics, which is basically a what you call reverse engineering approach, right. So, you have the whole brain dynamics. Now, you are asking how do I do like something like simulated annealing or those kind of approach, where I can go from the brain dynamics to the GABA glutamate concentration parameter space and see how does it changing as you become older, right. What is the optimal value for the GABA and the glutamate concentration. So, once you identify that, you can actually simulate a quantity which is called Euclidean distance. So, you have the experimental functional connectivity, you have the simulated functional connectivity, and element wise you can find out the Euclidean distance between those points, which is also called Frobenius norm in mathematics, and you can calculate the FC distance between empirical default mode network and simulated default mode networks to see whether they have some kind of agreement at different glutamate and GABA concentrations. So, you can do it for 3 different cases. The first case where you have the experimental data, second you have the simulated data for optimal glutamate value, then you have a second scenario where you have the high glutamate value, and you have a third scenario where you have a low glutamate value. This is just shown for the specific regions of interest or brain areas. This is the same plot which is shown over here in the connectivity matrix. What it turns out that optimal glutamate value in millimolar concentration is the best predictor of the experimental data, and when you are operating at high glutamate regime or low glutamate regime, then it fails to actually produce the experimental data. It much like a scenario like pathological aging, right, when you are dealing with low glutamate or high glutamate values. Okay? Yeah. So, key takeaways, and I will end my talk over there, is we could predict the age that negatively correlates with glutamatergic regulations, which was reduced with healthy aging. This is in concurrence with earlier evidence put forward by Davis and others, and also by Peter Bandettini, Suri and who measured actually MRS in vivo. And increased excitability in older adults was speculated to underlay the increased prevalence of certain neurological disorder, including epilepsy, because if the neurons become hyper excited, there is a chance of getting epilepsy easily at that particular older age. They become more vulnerable. And one crucial observation from theoretical approach that we have, or theoretical framework that we have proposed, is that the age related alteration are different in GABA than glutamate, even though we varied GABA glutamate concentration homogeneously across brain areas, and that remains an open question, because in ideal scenario the GABA glutamate ratios are also heterogeneously distributed across multiple brain areas. Okay? So, it was a very simplified picture that we are providing, but we are understanding or getting a mechanistic insight about how does the brain dynamics and functional connectivity reorganization specifically alters at the face of structural loss. Recently, in fact, there was a study by Gonen who actually looked at the MRS on glutamate and GABA of the posterior cingulate cortex and precuneus, which are the primary anchoring regions of the default mode networks, much like the previous slide that I have showed, documented that the glutamate and GABA did not significantly correlate with age, but their ratio actually did. Okay? Whatever may be the case, the proposed framework can be a potentially clinical tool besides magnetic resonance spectroscopy in studying patient data against healthy control, keeping GABA glutamate concentration as a primary constituent, and we are yet to uncover more potential of this MDMF model as to what it can do in terms of giving us potential insight in other neurological disorders. So, thank you for your attention.